All right, we'll pass 2, so we will get started. First, just to talk about the test, which we have on Friday. Like normal, it will open at midnight on Friday and close at midnight 24 hours from there. So you have all day Friday to take it. The material that's on the test is up till Monday. So anything that we go over today will not be on the test. Rather, today is the start of test 4. I have the rough draft of the test. So far, I'm up to 25 questions, with like 18 of those being multiple choice or some kind of you don't need to type in an answer type of question. But that might change slightly when I go over it again on Thursday, when I do my final run through. But you should expect more multiple choice on this test compared to your previous test. No math so far, and looking through the material, there's not really any math questions. Math will come up today, maybe, if we get to it. But test 3, we won't have to be dealing with any of those pesky equations we love so much. So anyone have any questions they want to ask about test 3? Not so much about the material. If you have questions about the material, email me. We can set up a meeting there. But anything just about test 3, like how is it going to run or anything like that, while I set up the PowerPoint here. Will we have a similar question to number 6 on the practice exam? Let me go look up the practice exam to see what number 6 says. For alpha, beta linkages, how do you know it's pointing up or pointing down? You have to look at the you have to look at the one, you have to look at the CH2OH group on carbon 6 and see what direction that is pointing to. And then you need to find the former carbonyl carbon and see which direction that OH is pointing to. So are they both pointing up? Are they both pointing down? If they're pointing in the same direction, that's beta. If they're not, that's alpha. So a good way to like test yourself on those is look at the questions we did in class and look at the practice tests. Practice exam, question 6. Question 6. Where are glucosaminoglycans usually found in the body? Relate their structure to their function? That type of question? That type of question is definitely fair game, because we did, briefly talk about it. And the answer for that is glucosaminoglycans are usually in connective tissue in joints because they're very negatively charged, and they're large, so they form large gels. You mean question 4? That makes more sense because question 6 seems pretty obvious. No, describe how the structure of potassium voltage gauges channels are related to their function. We have not covered that yet. So this just shows you that different semesters go at different speeds. That will be on test 4, more likely than not. But yep, if you see stuff on like the test that you're like, I have never heard about this at all, it's because we haven't covered it yet. So nope, not question 4. There are different models that we use, like the cherry conformation with wedge and dots. How can we tell on those? I'll do that really quick. Let me just share. current slide. Okay, so with the alpha beta stuff, I'm just going to draw a really crappy ring here. So there's like a couple different ways we can do it. If it's a wedge, right, that means it's coming at you. So the CH2OH and the OH here, they're both wedges, so they're both coming at me, so that's beta. Dash means it's going away from you. So if this was a dash instead and this was a wedge, that would be alpha. And then for the lines, you have like two different lines. One's kind of like going like that, and the other one would be like pointing straight up. So you're just looking to see if the direction is like the same. They will usually try to do that where, this pointing up means that I'm going straight up, and this kind of like slanted down line is trying to show that it's going behind. So those are the different types of like things you would see structurally or drawing wise. All right, but we do need to continue material since, like I said, on Monday, we moved everything back already. So we're going to cover Monday's PowerPoint today. And where we ended up on Monday is that we were talking about the translocon and how proteins that are destined to be either membrane proteins or excreted outside the cell, the first thing they have to do is that they have to be transported to the ER. Hint, hint, make sure you know how that process works. And we're going to continue right up after that. So after the proteins have been to the ER, the next place that these proteins go, if they're transmembrane proteins, if they're going to be secreted, or if they're lysosome proteins, they're going to go to the Golgi. So after being, folded in the ER, their next step on their adventure is to go to the Golgi apparatus. And just to briefly talk about the Golgi apparatus, we have 3 to 6 different membrane sacs called cisternae. And there's different ways we can move, but the way that proteins enter, so here's the ER, this yellow stuff is our ER. Right next to the ER is our cis, and where we leave, that's the trans cisternae. So we have cis, medial, and trans cisternae. Okay. So there are different ways that a protein can be moved through the Golgi. One way is that they can form little vesicles, which is being shown here, and they kind of like bud off, and then they reattach to a different part of the Golgi. So if you are budding off, that's called Retrograde Transport. So if I bud off one compartment and then I go to a different compartment. While if I travel in my membrane sac, right, and I just travel along with the sac as it naturally progresses, because these membranes aren't static. The cis will eventually become the medial, and the medial will eventually become the trans. It's like a conveyor belt. So if I'm just in the cis and I get carried all the way to the trans without like budding off, that's called cisternal progression. So that's cisternal progression, or it's called cisternal maturation. And it is possible that you can go backwards. So if you go backwards, which is actually being shown like here, so I bud off and I'm going backwards now, that's called retrograde transport. So that's retrograde transport. And the whole point of being in the Golgi is that you're going to go through any further modifications. And then you're going to be packed into this nice little vesicle where you can butt off and go to the plasma membrane, and you're either inserted into the plasma membrane if you're a transmembrane protein, or you'll be secreted as shown here with these proteins. They're being secreted from the cell. So any questions about our terminology for the Golgi apparatus? Yeah, the Golgi can do like other post-translational modifications of different proteins. Can you shorten the directions? I'm not quite sure what you're asking there. I mean, I can go through it really quick again. If you butt off and you're going from cis medial to trans, that's antrograde. If you're just traveling normally without butting off, that's cisternal maturation. And if you're going in reverse, that's retrograde. I'm not quite sure what you mean by shorten. Like, can you take a shortcut? Oh, you just wanted me to repeat it again? Okay. So let's see how well you listen to that. So if I go from one compartment to another, what is that called? And I have a quick poll here, just so we can have everybody answer, so everybody can be a part of our process here. I understand this question is a little vague because, well, I'll explain why it's a little vague in a second. Or is it vague? No, it's not. It was vague last semester. I made it actually not vague this semester. All right. So let's end this poll. So I realized that the poll question and the question I have on the PowerPoint are different because the one in the poll is the one I had originally, and then I made it on the PowerPoint to be more specific and forgot to change it. So aren't cisternal progression and maturation the same? It's cisternal maturation. It's just not maturation. It would be the same if I put the word cisternal in front of it. All right. So the correct answer, which I was looking for, is that proteins are passed by budding from one Golgi compartment to the next. That's called antrograde. So that would be D. But in the actual poll, I didn't say that you're budding off. So if in the poll, you just read the poll question, B would also be acceptable if I did not say budding. Oh yeah, so I might have just updated that on it's possible that I didn't upload the budding part to Blackboard because that's something I changed very last minute. So that's why I said, you know, this question wasn't as specific as I would like to do. I mean, if I did not say budding, D and B are both right because both antrograde and cisternal move in successive compartments. The difference is what I was trying to get at, is that antrograde does it by budding off, and cisternal, you just kind of ride along in your little vehicle. Can I go over maturation, cisternal progression? Sure. There's 3 compartments of the Golgi: cis, medial, and trans. If you're a protein, sure. That's my protein, Mr. Smiley Face. What you're going to do is you're just going to follow along, because eventually the cis is going to become the medial is going to become the trans, and the trans kind of like buds off. So if you're doing this, if you're doing maturation, cisternal progression, you just never leave your little hot pocket, your little Golgi pocket that you enter in. So you enter through the cis, because the ER is up here, right? So you enter the cis and you just ride along until you become medial. And then you just ride along until you become trans, because the Golgi will naturally change like that. That's why I said it was like a conveyor belt. If I go back to my previous slide, like these naturally move, because you can see at the end here, the trans are budding off. So the cis eventually becomes a medial, medial eventually becomes a trans, trans eventually becomes secretory vesicles. And if you are just in here and you never leave, eventually you're going to become trans. If you travel that way, that is cisternal progression or cisternal maturation. All right. So let's say that you are, what is maturation alone? Nothing. That was a fake answer. So let's say that you are a transmembrane protein, right? How do you actually get inserted into the plasma membrane? This diagram shows us how, right? So when you come off the Golgi, when you leave the trans Golgi, you are going to leave in a vesicle. And these vesicles, they're not just lipids, they're going to be coated with proteins, which we'll get to in a second. But what happens is that this vesicle, and this picture is kind of hard to understand going on the first time you look at it. So it's like step 1, step 2, step 3. What happens is that this vesicle will go and fuse with the plasma membrane. The part of the protein that's inside the vesicle, the inside is called the lumen, that's going to point outside of the cell. So depending on how you want your protein to look like, do you want your protein to be pointing inside the cell or outside the cell? You have to put it in correctly in the vesicle. Whatever's in the lumen will point outside. So what happens is that your vesicle comes near and it fuses with your plasma membrane. That's what's being shown here. This used to be the vesicle. It's just fusing now. And so your plasma membrane gets a little bit stretched out when you add this vesicle. And finally, you're just going to have plasma membrane and your proteins attached there. That's also the basic mechanism. If you want to completely expel something from the cell, you will put it in a vesicle. Whatever's inside the vesicle, when it fuses to the membrane, anything that's in the inner area will just be expelled outside the cell. What's the difference between maturation and progression? Nothing. It's the same thing. Two words that mean the same thing. That's why in the previous slide I have, just to go back there really quick, cisternal progression / maturation. Different people call it different things. All right, any questions about our vesicle process here? Yes, retrograde is going backwards. You either go trans to medial or medial to cis, or trans to cis. You just go backwards in the process. That is what retrograde is. Medial grade. I don't know. I did not talk about medial grade. I don't think medial grade would be a fake answer. Yes. Not every answer I give you is real. All right. So moving on. So we know of 3 different types of proteins that will coat vesicles. And depending what protein is coating your vesicle, it will tell you what direction your vesicle is traveling. So first we have Clathrin. Clathrin goes from Golgi to plasma membrane. Then we have COP1. COP1 will go to Golgi to ER. So let me actually draw a little picture here. So here we have our ER. Here's our Golgi. And then all the way out here is our plasma membrane. So if my vesicle is going from Golgi to plasma membrane, that is Clathrin. If my Golgi is going back to ER, that is COP1, also known as codomer. Right, and that's just because some proteins have to stay in the ER. They are ER-specific proteins, but just naturally, proteins that are in the ER will be transported to the Golgi. So a mistake could happen in which a protein that is supposed to stay in the ER accidentally goes to the Golgi. So we need to return them back to their home. We need to return them back to the ER. And so we have COP1 coded vesicles to do that. And what COP2 does, COP2 will go the reverse direction. That's why they're named like that. So COP2 will take proteins from the ER into the Golgi. And since COP2 is going that direction, they need to return back to the ER. And so what COP2 will do is that they will travel back in COP1 vesicles. And Clathrin, COP1, COP2, if that's my vesicle that I just drew, these proteins are just on the outside of it, right? Coating it, helping to form this vesicle. That's what I'm talking about. And the proteins that are on outside determine which way we're going. Questions about that idea? All right. Yes, Clathrin, COP1, COP2 are all proteins. The vesicles themselves aren't proteins. The vesicles are a mini lipid bilayer, but it's the things that I just talked about are proteins that coat the vesicles. So that's like what they're doing, right? Because these proteins on the outside, they can interact with certain receptors. And when you bind this receptor, you'll be taken in. So for example, ER, on the outside of ER, you have proteins that can recognize COP1. And so they'll bind to like COP1 and say, okay, yeah, you're cleared to come into the ER. Golgi can bind COP2. So it'll say, oh, this is a vesicle with COP2, that must be coming to me. Plasma membrane can recognize this Clathrin. Oh, you're Clathrin? Okay, you must be coming to the plasma membrane. Please come in. So yeah, they are recognized by receptors at their final location. All right. So let's just look at one of these proteins here. Whoops. And so this is Clathrin, and they're made from a protein that are known as triskelions. Basically, the way I think of it is like a three skeleton, triskelion. The picture is down here in the blue. And you got 3 heavy chains, and each heavy chain is bound to 2 light chains. So it's not exactly shown that well in the picture, but here we have heavy chains in the blue and your light chains in the yellow. Now, when you do not need to make a vesicle, the light chains will be bound to the heavy chains, and the light chains are a way to prevent polymerization. And so we can see in this image down here, we have a bunch of Clathrin that is not forming a vesicle, and that means our light chains are bound. However, when you need to form a vesicle, the light chains are removed, and then you can form this polyhedral cage. And the cool thing about this cage is that it can form different sizes and it can form different shapes based on how the Clathrin polymerizes, which is really good because these vesicles, they're almost like shipping boxes, right? And so you are going to be transporting different size products or different size proteins. So you need to be able to be flexible with your vesicle to accommodate all the different proteins that you are shipping. Now, just looking at our other image here in the middle, since we're talking about transportation, and let me rewrite the words down here. So this is lower H+ concentration, lower pH, peptide releases. And what we're looking at this middle picture is transportation between the ER and the Golgi. So here at our we start in the rough ER, right? And these blue and like purple proteins, these are ER resident proteins. They are proteins that have to stay in the ER. They work in the ER. Their job is at the ER. And sometimes by accident, these proteins will go into a COP2 vesicle and they'll be transported to the Golgi. Now, these proteins have a C-terminus sequence, a C-terminus tag rather, that tells the cell, hey, this protein must stay in the ER. For mammals, the C-terminal sequence is usually KDEL, lysine, aspartic acid, glutamic acid, leucine. In yeast, they use HDEL. What can also be in mammals is lysine, lysine, and then 2 random proteins, and then or lysine, 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 3 random proteins. Right. So this is at the C-terminus of proteins that must stay in the ER. If you get accidentally transported to the Golgi and you belong in the ER, the way you get sent back is inside the Golgi, we have proteins whose job it is to just bind this KDEL sequence. So if there's a KDEL sequence floating around in the Golgi, this receptor will bind to it and more or less capture your ER soluble protein. Then these COP1 vesicles will take in your KDEL receptor and form a vesicle around them, go back to the ER, and they will deposit your ER resident proteins. And you notice COP2, we also have the receptor here. So we want to recycle our stuff, right? We don't want to have our KDEL receptor be used once and then be worthless. And plus, if it was in our ER, it would just bind a ton of stuff. So we want to get our KDEL receptor out of there. So we'll just send it along in a COP2 vesicle to go back to its home in the Golgi. So that's how the the cell can tell if a ER resident protein accidentally left the ER. It's all because of the C-terminus sequence, and they can recognize that, bind it, and ship it back. And here are, this slide is really just going over our three main types of transport proteins, and just in very, very general terms, how they work. So any questions about the information presented on this slide? If not, we'll move on. Okay, so I have some questions to test our understanding. And since these are multiple choice, let me bring up another poll here. Okay, so basically this question is, what is true about Clathrin? Well, all I can say is if it feels like a trick, always trust your gut. Usually on problems on tests, your first intuition is right. Your first thought is going to be right more times it is than it's going to be wrong. Okay. So let's see here. Clathrin. What do they do? Well, yeah, they transport. I'll share the results. Yeah. So the correct answer is B. They transport proteins from the Golgi to the plasma membrane. That's like what I said 2 slides ago. That is their purpose. That's what they do. So that's a good review on Clathrin. Hopefully that's inside your head a little bit better now that you had to think about it and see the correct answer. But it is B. They go from Golgi to plasma. Okay. How about ER soluble proteins now? So I should have another poll for this. So let's take a look. Yeah, this should be multiple answer, I think. If it's not, I messed up. But anyways, which of the following are probably ER proteins based on their C-terminal residues? And if I didn't make a multiple answer, I'll just let you know right now there are multiple answers. So just select one then and write down what you think the other ones are. All right, give like 10 more seconds. Yeah. Seems we did pretty well at least getting one of these. So I'm going to go by them one by one. Well, actually, I lied. So the sequences we're looking for, KDEL, and in yeast says HDEL. Or you can be KKXX or KKKXXX. That's the sequence we're looking for. So not A, yes B, not C, yes D, not E, not F, yes G. So the possible answers for these are B, D, and G. Those are possible or probable ER proteins based on their C-terminus sequence. And really, we're just looking for these sequences and at the C-terminus, and that will tell us if it's an ER resident protein. Because remember, in the Golgi, we have receptors that look for these sequences and will bind them and send them back to the ER on these COP1 vesicles. So any questions about that, those ideas? All right. So how do vesicles actually fuse with the plasma membrane? And to actually understand this, think of the problem, right? The plasma membrane and vesicles are made out of lipid bilayers. The head of a lipid bilayer is usually negative. They're negatively charged. And so if I want to bring a vesicle and put it next to a membrane, I'm putting a negative next to a negative. They're going to repel each other. They don't want, the vesicle and the plasma membrane don't want to come in contact with each other. So to help this, we need other proteins to help with the fusion of vesicle and membrane. And we're just going to look at one of these proteins, and they're called snares. And I'll just say straight off, the process I'm about to describe is very, very simplified. Because if I tried to do this with just snares inside a test tube, it takes about 30 to 40 minutes for my vesicle and my plasma membrane to fuse. While inside of a cell, this takes 0.3 milliseconds, right? So there are a lot of other proteins that we probably don't know about involved in this process, but I'm just going to talk about snares as one protein that does it. So there are two types of snares. You have R snares and Q snares. R snare will be on your vesicle. Q snare will be on your membrane. And what will happen when your vesicle and your membrane come together is that these snares will start to wind against each other, almost like twirling rope together. And what will happen is that when your snares are twisting like that, your vesicle and your membrane gets closer and closer, and your plasma membrane at that location starts to bubble out a little bit. Then through some mechanism that not being explained here, you will lose some lipids. These lipids will just go away. This is what's called hemifusion or hemifusion, where you have like you're halfway fused between your vesicle and your bilayer. So what will happen is that your zipping will continue. Your snares will continue to wrap around each other. And this fusion will continue until you wrap so tight, you form a little pore, right? And so if you are just transporting something to be secreted, when you have this pore, the proteins are free to leave. If you had a transmembrane protein, though, like if my protein was right here, now my protein is part of the membrane, because you are more or less breaking the vesicle and having it form with the plasma membrane. And this continues until your membrane is now a little bit bigger than where we started from. Your vesicle is completely gone, and your vesicle is now part of the plasma membrane. But think of the snares almost like kind of like zip ties or some other piece of string that when you wind it around each other, you wind, it'll get more and more tense, right? And it's literally dragging this vesicle and this plasma membrane together to force them to interact and to force them to become one. So that is how we can overcome not only the electrostatic repulsion of the negatively charged lipids, but also the desolvation penalty. What I mean by that, so the word I just said was like desolvation penalty. This vesicle and this membrane are surrounded by water. When you bring them together, you have to kick the water out because it has to be lipid touching lipid, and that's unfavorable. So they don't want to do that. They don't want to kick the water out. And so these snares help to overcome that. And like I said, there are a lot of other proteins that have to be involved in this. We're just talking about the snares basically here. Any questions about that information? How do the snares attach to the vesicle membrane to begin with? That's a good question that I actually don't know the answer to. I've never looked that up, but my guess would be that it's the same way that a transmembrane protein is transported to the plasma membrane. That, like in the Golgi, right? You're just going to have these snares being made. They will go to the ER and then go to the Golgi, and these snares will be inserted into the plasma membrane. How the Q snares get there in the first place, maybe they're interacting with other proteins, right? So that's part of the mystery that I do not know of. That's a good question. Unfortunately, I cannot answer that sufficiently. What makes the snares want to keep wrapping like that? Another good question. Are snares, what amino acid is arg? That is arginine, like positive, right? Q snares, what amino acid is Q? Glutamine. They have C double bond O's. It's going to be some hydrogen bonding electrostatic interactions that make the amino acids want to associate with each other. So it's based on amino acid interactions. If the outside is negative on both the vesicle and the membrane, how did the vesicle get close enough for the snares to interact? Because they're like, the snares are like little arms, right? And so the arms can like reach out and make that first contact without, because right here, when they're first making contact, you still have water in there, right? So water is going to be interacting. And so it's okay that they're this close because basically, the reason why salt dissolves in water is because water is very good at screening charges. That is, if I'm a negative charge and I'm surrounded by water, I'm okay. I don't know about any other positive charges around me that well. I don't know about any other negative charges. I'm happy. So when the vesicle and the plasma membrane are close enough for the snares to interact, there's still water there. So the vesicles really don't know like they're gonna hit another negative charge. They're being screened. They're like blindfolded until the snares start twisting, and then the water is forced out of the way. Then it's like, oh no, I'm coming into another negative charge, abort. But they're already tied in, so it just gets tighter as your snares interact. And like I said, there are other proteins that are doing this, just not snares. There are other proteins that are helping bringing these two together. But yeah, and the other proteins like the Clathrin could also be helping with this initial interaction with the plasma membrane. Yeah, that question has a lot to do with biophysics and a lot to do with dielectric constants and how far you can see charges in water. So there's a lot of math to that actual answer, but that's the shorthand version of it. Yeah, that is biophysics right there. And understanding the snares itself. Yeah, I guess it would be fun if you're into that type of stuff. I'm more into proteins than lipids myself, but you know, each of their own. All right. So, which of the following lets us know or tells us why snares are useful? Hopefully, I made this question correctly. I'm guessing I didn't. I did not. This one also has multiple answers. So, see if you can at least get one answer, and then see if you can get the other multiple answers by writing it down on your piece of paper. Or I guess if you're not using a piece of paper, just think about it. Give about 10 more seconds and then move on to like one more slide here. All right. So the correct answers here are 3 and 4, right? So snares don't do post-translational processing. Post-translational processing is done in the ER and the Golgi for these proteins. Snares have nothing to do with that. All snares do are bring vesicle and plasma membrane together. Secretory pathway is getting a protein into the ER to begin with, which you're being tested on Friday, so hopefully you know that. So snares don't do that. Remember, that's like the SRP and the SRP receptor. But they do help to overcome electrostatic repulsion, so 3, and they help to anchor vesicles, vesicle to the plasma membrane. So, and they could also do vesicle to vesicle. They basically, anytime you have lipid bilayers that want to come together for some reason, snares are going to be involved in mammals for that to happen. Questions about that? Let me just see here. Influenza and thermodynamics. Okay, so I'm actually going to call it there because it is 2:50. So Friday we have the test. So if you do have questions about that, please let me know via email, set up a meeting, whatever. Make sure you do the old tests online. Keep in mind that what you'll find on test 3 does not necessarily correlate to all the material. So make sure you look at test 2 as well. Test 2 will probably have some information on there that will be relevant to our test 3. Just because, like I said, the different speeds of different semesters, different material ends up as slightly different practice tests. Yeah, make sure you go over the reading guides. Make sure you go back to the questions in the PowerPoint to just test your understanding. But yeah, if you can do all that, I don't think this test is going to be a problem if you do all that preparing. So I said at the beginning, 25 questions, about 18 of them multiple choice. I think that was the big thing. Yeah, honestly, my brain's a little foggy since, against my better judgment, I was up way too late last night. But that's the main point. All right. If there's no more other questions, yeah, email me if you do have questions and want me to go over anything one-on-one, or if you just want to shoot me a short question by email, I'm happy to do that too. Otherwise, good luck. Test opens that's the other thing. Test opens at 12 a.m. on Friday, closes at 11:59 p.m. I said 12 p.m. in Gen Chem, and everybody yelled at me saying I forget. 23:59. It's going to open at 00:00 and close at 23:59. No confusion there. All right. Take care, everybody. Have a good one.